Okay, so if you remember from last week, we talked about plant anatomy, which is really looking and identifying the structures of the plant, whether it be tissues or organs or anything else like that. Well, this week we're talking about plant physiology. In physiology, we're talking about the function of those structures that we covered last class. Now, plants can undergo a lot of different physiological processes, so we're really just going to focus on transport in this video. And with plant transport, there's really two major types. We have transpiration and translocation. And there's key differences between the two of them. However, both of them are going to happen in the vascular system, so that xylem and phloem that we covered last time. Okay, so just talking about some of these differences. With transpiration, it is going to transport water and nutrients, and this is going to happen in one direction. So water and nutrients are going to come in through the root system, go up the plant through xylem, and then leave through the leaves through evaporation. And this process is going to be passive, meaning it requires no energy from the plant for this to work. And by energy, we mean ATP. So the plant does not need to exert any ATP for this to function. Unlike translocation, which is the transport of carbohydrates. And by carbohydrates, we mean sugars. This is going to be an active process, meaning the plant needs to use ATP for this to work. Now, these carbohydrates are being made through photosynthesis. So some leaves are going to undergo photosynthesis and make these sugars, and these leaves are going to make more sugars than they need. So we call these leaves that are undergoing photosynthesis and making this surplus of sugars, we call them sources. However, cells all across this plant need those carbohydrates. Plants, just like most eukaryotes have mitochondria, they need carbohydrates in order to create more ATP to do work. So this plant needs to pass these extra sugars to any of the cells that may need it. So this needs to go all different directions. And what you're going to see is this is an active process. This is actually going to cost ATP for this to function. Now, the leaves that we are creating these sugars, we call them sources, and all the tissue that need these sugars but don't make enough sugars from themselves are called sinks. So again, transpiration using xylem, and it's one direction and it's passive, while translocation is using phloem and it's multiple directions and it's active. Okay, so it's also important to understand where we find this vascular tissue and how it's arranged. So here we're just looking at a cross section of a root right here, and towards the center of the roots, this is a center of a dicot, but if you look towards the center, that's where you're going to find something called the vascular bundle. And the vascular bundle really is a bundle of xylem and phloem. Now this is a cross section, so you could view it under a microscope. So we're looking at the kind of the top of the vascular bundle here, but if we were able to take a longitudinal section, this is more what we'd see. So with these, the largest cells are going to be your xylem, while the smaller cells next to it are going to be the phloem. So I just have it kind of drawn out here. I know it's a pretty rudimentary drawing, but hopefully it'll do the job. All right, with xylem and phloem, they're usually next to each other. I'm going to start with xylem first. Xylem is usually dead at maturity. Really, all xylem is the cell walls that were left behind by the cells that were growing here. But by the time they reach maturity, the cells die and they just leave behind their cell walls to make tubes. And there's two major cells to xylem. There are vessel elements, and then we have tracheids. Now compare that to phloem. Phloem's actually alive and needs to be alive for it to function properly. And that's because things need to move all different directions and needs to be able to use ATP to make that happen. So the cells need to be alive for that to occur. And there's two major cells to phloem. There's sieve tube cells or sieve tube members. These are going to make up the tubes that are going to be running through the entire plant. They are separated by sieve tube plates. And then we have companion cells next to them. Okay, so with the companion cells, these are loaded with mitochondria. And this is going to make all the ATP and donate it to the sieve tubes to use, because the sieve tubes have lost most of their organelles in order to create open space to maximize transport. So these sieve tubes, again, lost everything. They lost their organelles, are just there to make tubes. And the companion cells are really kind of picking up the slack by having extra mitochondria to donate ATP to these sieve tubes. Okay, so the first process we're going to go through is transpiration. And remember, transpiration is one directional and it's passive. So it's going in through roots, up through the xylem, and then out through the leaves as evaporation. So we're going to talk about why transpiration is passive, why it doesn't require ATP to function. And that has a lot to do with the properties of water and the vascular system. All right, so to fully understand this, we have to see it happen in leaves, look at the vascular tissue in leaves, look at the vascular tissue in the rest of the plant, and then how water is entering the root, because all of these are related to each other. Okay, so we're going to start with the leaf here. So this is a cross section of a dicot leaf. And here is your vascular bundle, your xylem and phloem. And remember, water is going to come through the phloem. So with dicot leaves, we have a layer called spongy mesophyll, which is this very airy tissue that is right below the leaf. So you see all this open cavernous space here, and it's all leading to something called stomata. And stomata are pores or openings on the outside or on the lower dermal layer of the leaf that allows gas exchange or gas to come in through the stomata and reach this spongy mesophyll or this open space in the spongy mesophyll. So this is where water is going to be evaporated mostly, is from the stomata. And stomata are guarded by something called guard cells. So guard cells can open and close the stomata as needed. Okay, so let's talk about why this is a passive process. All right, so all this open space is going to have water molecules in it. And the air that the plant is in is going to have less water molecules in it. And so water has a tendency to want to equalize where there is less water. So since there is more water here than outside the leaf, water molecules are naturally going to want to leave through the stomata and enter the air and evaporate into the air, just simply because there's more condensed water here than there is out here, because water wants to equalize itself. Well, water has another property called cohesion. Because water molecules are polar, they have a tendency to stick to one another. So every time one of these water molecules leaves the leaf, it's going to pull another water molecule with it through cohesion. And so you're creating this chain effect. Every time a water molecule leaves, it pulls another one with it, and you're going to have this chain effect all through the xylem and then eventually down through the entire plant, where each water molecule is going to pull the next water molecule up, creating this constant chain of going from bottom to top and then leaving as evaporation through the stomata. Okay, so evaporation is kind of explaining the unidirection of transpiration, but it doesn't really explain how it's not being overcome by gravity. All right, so this is a long way for water molecules to go, and cohesion alone is not going to allow this to happen. Well, evaporation with cohesion alone is not going to allow this to happen. And it really is up to another property that water has called adhesion. So adhesion is water's tendency to stick to other objects. So if this is our xylem running up the tree, we have water in here that has a tendency to stick to the sides of the xylem. Just like if you stuck a straw in some liquid, the straw is going to have a little bit of water higher than the surface of the cup that you stuck the straw in. And that's because of this adhesion that water displays. And this creates something called a capillary effect. So water naturally kind of wants to climb up these thin tubes through capillary action. And this is what's going to help prevent gravity from taking over. So if xylem gets too big, if xylem is too wide, this whole system shuts down. The adhesive forces of water is not strong enough to fight gravity with too big of xylem. So xylem needs to be the right size for capillary action to work. All right, and then the last piece of the puzzle comes down with the roots. So water needs to enter the roots from the soil, the surrounding soil. And this happens in a couple of different ways, but the majority of the way is from taking up nutrients. So there is nutrients in the soil, and the plant is going to take in nutrients usually from some sort of active process. So ATP is needed to bring in nutrients. So you're bringing in these nutrients into the cells of the root. Okay, so what's happening here when we build up the concentration of these solutes inside the root? So now there's higher solute concentration in the root than there is in the soil. Well, if you remember from osmosis, water is going to want to try to equalize these two things. So osmosis wants to try to make sure there's an equal concentration on both sides of the membranes of the cells that are in this root. Since there is more solute inside the root now, water is going to pass the membranes of the cells of the root and try to equalize the concentration of these solutes. So water is going to start moving inwards through osmosis. So water entering the root is actually a passive process, even though it takes active pumping of these solutes into the root for it to occur. And so here's our complete system. The plant is doing this all the time. So it's usually storing water and building up water in the root. And then during the day, it could open its stomata and allow transpiration to occur, bringing water up the rest of the plant, making sure every other cell gets some water. So even though it takes some energy to bring water into the root, moving the water up the plant, fighting gravity, and making sure all the other parts of the plant get water is a passive process. The only thing that needs to happen is that the guard cells need to open the stomata, and this will happen. This will occur. Okay, so that is transpiration. Okay, so next we're going to talk about translocation. And remember, translocation is an active process that can go any direction in the plant. And really what we're moving here is carbohydrates, so sugars. And with translocation, we're going to go from a source, the place that is creating sugars, and go to a sink, the place that needs sugars. All right, so for it to be not unidirectional like this, to go multiple directions, it needs to be an active process. So ATP is going to be used in some way. So here I just redrew the vascular bundle. Here, so let's say this is xylem here. This is going to be our sieve tube. And this is going to be our companion cell. Okay, so next to the companion cell, you're going to find the leaf cells that are making all the sugars that are going to be transported through translocation. So just imagine the leaf cells are over here somewhere and they're loaded with sugars. And the sugar we're talking about specifically is sucrose. So even though glucose is what's being made through photosynthesis, plants actually turn it into sucrose for transportation. So let's say these are our sucrose molecules right here. Okay, so how are we going to get these sucrose molecules into the companion cell and then eventually down the sieve tubes? And we're going to do this using something called a proton pump. Now, what a proton pump is a protein that is inside the membrane of the companion cell, and it's going to use ATP. So it needs energy to work. And so what it's going to do is take this ATP and use the energy from the ATP to force hydrogens or protons across the membrane of the companion cell to outside the companion cell. And what we're doing by using this proton pump is increasing the concentration of hydrogens outside the companion cell. And we're creating this concentration gradient from the outside to the inside of the cell. But ATP is being used for this to happen. And then once we have this high concentration of hydrogens out here, remember, hydrogens are going to want to go down their concentration gradient back into the cell. However, they can't pass this membrane easily. So instead, they're going to need to use another protein to get back in. And what they're going to use is something called a cotransporter. Now, everything in the name tells you what it's going to do. It co-transports things. So it's going to transport two things. It's going to transport the hydrogen down its concentration gradient using that energy that's naturally there from this concentration difference. And it's also going to pull in a sucrose. Now, this protein won't work unless both the sucrose and the hydrogen are attached to it at the same time. So it's going to have to pull both in simultaneously. Then once it does that, we're going to start building sucrose concentrations inside the companion cell, just from this concentration gradient that is established using the ATP and this proton pump. Now, once we have a high concentration of sucrose inside the companion cell, it's going to want to go down its concentration gradient into the sieve tube. And it's able to do that because the cell membrane between the sieve tube and the companion cell is perforated. So they're going down their concentration gradient and loading the sieve tube. Okay, but we still need to get this sucrose down the sieve tubes to the sinks where they need to go. So this is where xylem comes back in to play. So remember, xylem has all these water molecules in it, and they're going one direction due to transpiration. Well, since there's this high concentration of sucrose in the sieve tube, and there's no sucrose in the xylem, The water molecules are going to pass the membrane through osmosis to try to equalize the concentrations inside the sieve tube. So water's entering the sieve tube trying to equalize the concentration of sucrose. And we say water is going down its water potential. There's a high potential here where there's no sucrose, and there's a low potential here where there's a lot of sucrose. And it's trying to equalize this concentration. And what happens here is as you increase the volume in the sieve tube, you have this force pushing outwards. But there's a cell wall here preventing the cell from expanding too much. So you actually have a force pushing back from the cell wall. And you're increasing pressure here as volume increases. So there's a pressure buildup here, and we call this a turgor pressure. And this pressure is going to force these molecules down the sieve tubes, pushing them outwards. So let's say there's a cell here that's acting as a sink and then needs to collect the sucrose, or it needs sucrose. Well, now we have these sucrose molecules and water being pulled down the sieve tube. And since there's no sucrose in this sink, because it needs sucrose, and there's a high sucrose in the sieve tube out here by the cell, sucrose is again going to go down its concentration gradient and enter into the cell. So we're losing sucrose from the sieve tube now because it's all entering this sink, the cell that needs it. So now we're just left with water here. And if you remember in the xylem, it was also carrying nutrients like phosphorus and nitrogen. So since there's a high concentration of phosphorus and nitrogen in the xylem, and there's no phosphorus and nitrogen in the sieve tube, water again is going to enter the xylem through osmosis, where it can then be carried up through transpiration again. Okay, so just to recap all this, we are going from source to sink with the sucrose. With the companion cell, we're establishing a concentration gradient of protons using a proton pump and ATP. A co-transporter is then going to pull a sucrose and proton at the same time back into the companion cell, going down that proton's concentration gradient. Then we're going to have a buildup of sucrose in the companion cell that's then going to enter the sieve tube, going down its concentration gradient. Once there's a high concentration of sucrose in the sieve tube, water is going to enter from the xylem into the sieve tube through osmosis, creating a pressure as the volume builds up called a turgor pressure, and that turgor pressure is going to force these molecules down the sieve tube to where they need to go. Then at the bottom here, we have this sink, or the cell that needs sucrose. So it's going to take the sucrose from the sieve tube because sucrose is going to go down its concentration gradient. And then once that happens, we're left with water, which is going to enter xylem again through osmosis due to the nutrients that are in xylem. Okay, so that is everything I wanted to cover in this video. So that is going over translocation and transpiration.